Hello everyone. Welcome to the next lesson. So in this space we're going to cover basic sterile tissue culture techniques. We're going to go over a few principles of what we do when we're using the Biosafety Cabinet and other things within this tissue culture room. So basic things that you can find within the tissue culture room are always 70% ethanol to clean up the space that you're going to work in, tools to pipette volumes of media, solutions, and other such things, napkins to clean up as well, a vortexer, some pipette aids to help aspirate and dispense solutions, tubes that are sterile to work with, holders, water bath, a sink, and pipettes to assist with everything you got to do. We also have centrifuges that are either refrigerated or not, and they're all for the sole purpose of just helping us culture our cells and all the tissue we got growing at the moment. So I'm going to get started with some basic techniques. One thing to go over: this is a Biosafety Cabinet 2, which is suitable for growing cells. The airflow, I apologize for the drawing, but our printer's not working, but essentially the way we work is within the middle of the space, because the airflow comes in through the top and, as you see, gets pulled in through the sides, so our clean space is in the middle and then the rest of the air gets dispensed out. So there's always clean space within this middle, which is where we're going to work in. When you're working sterilely, you never come out of these realms. Never. Because the minute you come out of here, it's not considered sterile anymore. So you have to consider, when you're in a biosafety cabinet, to stay within the middle and never to stray out of their parameters. Okay, so let's get started. Okay, so here we have our controls for our light. So we have UV, which is UV light to kind of sterilize any bacteria that could be around there. You usually turn it on 20 minutes before we come in, but we turned it on already, so we don't need to do that. And what we're going to do is turn on our fluorescent bulb, and we're lit, and we're properly we can see properly to conduct our work. Okay, so we'll continue. So in our water bath set at 37 degrees, we have media that we're going to change our cells in, and hopefully what we're going to do is change our media so that our cells remain clean, bacteria and yeast free, free of any other pathogens, so that we can continue doing our experiments and continue growing and freezing and whatever we got to do in the lab. So one thing we'll start off is right out of the water bath, the solution feels warm enough to add onto the cells, and what we'll do is we'll just gently spray a little bit of overlay with 70% ethanol. It's okay if it drips down, but as long as it's gotten all over where the water has touched, and we're going to wipe it down clean. So wipe this down, and we're going to put it on our stand inside. Okay, so as I mentioned before, because of the airflow, this is our barrier. So to work sterilely, we're going to stay within this area. We're never going to come out here or stray too far out. So we're going to push everything out. We're going to keep our vents at vacuuming air out here and in here. So in here is clean. Okay, we're going to get comfortable and sit down. We're going to spray down our work area, make sure it's really as clean as possible. And we're going to wipe it down. Okay, so it's pretty much clean. And here we have our incubators set at 37 degrees at a certain concentration, 5% CO2, which our cells happily grow in. So we open, we get our cells out here. So as you can already see, we got these little dots are our cells. They're called organoids. They're growing in media. Media is supposed to be pinkish, orangey red, and this is a yellow, so it needs to be changed. So that's what we're going to do now. We're going to change it. So things that I need to change are my media, plate of cells. I have sterile pasture pipettes, glass pipettes in here, and we can just get started. All right, so we place our plate right in the middle, place our media bottle right next to it. We quickly open our container, grab a pasture pipette, close, holding one end and not the end that's going to touch our media, we're going to attach it to a vacuum. You make sure it's properly sealed so that it aspirates. You target the cells that are going to require the removal of its media, and we just place it in, and as you see, the vacuum just aspirates. Okay. So right now, all the media has been removed, and you currently see the matrigel dome containing our cells. These cells are grown in 3D, so they're in a jello-like substance that we call matrigel. All right, so we're gonna add a ml of media each. We're gonna remove a sealant that I put, and we're gonna grab our stripettes. And one thing I usually do is I usually put my pipette inside, sterile pipette, and I peel the top that's not gonna go into the media. So you have a tip that's gonna go in and a tip that's gonna go into our pipettement. So I hold it sturdy with a thumb and I peel it. It even has the instructions here. It says peel to open, and I peel it open and I use my thumb to remove the residual paper, and I attach it to the pipettement. Discard the paper. With another hand, I unscrew the media that we're going to feed our cells, and I take the amount that's needed per well. I have 5 wells, and I'm going to add a ml each, so I go up to 5 mils. I screw the cap back up. So now I angle the cap. So now the wells inside, the cells don't have media, and I need to eject the media inside, but it's covered right now. So what I do is I place the cover at an angle and I invert it, and I add the mil that it's needed. 1, let's kind of speed that up. 2, 3, 4. All right, trash in the biohazard charts. Media's changed. Cells should be happy. New food, new media, and we're pretty much done. It goes back into the 37 degrees where they're going to continue growing, and we'll take a look at it tomorrow as they grow. Okay, so that's pretty much what needs to be done for your cells to be clean, sterile, and I think if you follow the basic technique of never coming outside or opening your plate outside or opening your media outside, everything will remain sterile. Nothing that's unexpected will grow, and your cells will be clean and healthy enough to conduct an experiment. Okay, so that's pretty much about it. See you guys at the next lesson.